Hi, Professor Pandi, how are you? I hope I'm saying your name right. So the way to unmute is all the way on the bottom right. There's a little microphone symbol, and if you press that, you're unmuted, and then we can talk. Huh,
Yeah.
perfect.
Can you talk to me now?
Yes.
Okay.
Now
Okay.
I hear you. Thank you so
Okay.
much for coming. We really appreciate it so much.
Oh, it's my pleasure. Thank you so much. Yeah.
We have 10 more minutes, so you can just relax and while we wait. But
Sure.
yeah. And hi, Katie. Meet Katie. She is joining us from Australia, actually. So
Oh, wow. Hi, Katie.
she's a scientist too.
Okay.
Hi, it's Katie. Such a pleasure to have you here. Thank you, as always, Katerina, for bringing so many amazing scientists to the platform and everyone listening in. Yeah, please be patient while we have a few moments to bring people into the room. Please feel free to share the room on Clubhouse by pressing the share on the platform or on other platforms as well, because we want to have as many people as possible come and engage in this conversation. Really, really looking forward to hearing about your work, Professor, and already downloaded the link that's at the top of the room and excited for this conversation. Thank you.
Thank you so much.
Gabi joined us too. Hi, Gabi. Thank you so much for helping to organize this. Welcome.
Thank you.
Yeah, without Gabby, I cannot able to join this. She helped me a lot, So
Yeah, thank you to both of you. That's wonderful. I'm so glad she came too. So, yeah, thank you. So, it's the first time you're on Clubhouse, right, Professor?
yeah, I'm first time, but when I was registered, I saw my son is already there. So
Oh, your son. Oh, wonderful. he can chat here too now.
yeah, he's a neurologist,
Oh,
Uh,
cool.
yeah.
Yeah. Is he
So I
at the same?
I yeah, go ahead.
Oh, please go ahead. I'm sorry.
Yeah, he's a neurologist. He's working also. I mean, he's a MRI and brain. So I saw his name there too. So I told him.
Oh, nice.
So
Maybe he's coming too. Oh, if he's here, I'll bring him up too. Where we are is the stage, and then the other, if you see, followed by the speakers and others in the room, they're in the audience. So
oh,
if
okay,
you
okay,
speak, so people can share in the chat, but in order to speak, we have to bring people up here. So yeah, if you let me know, I'll bring him up. Is he at the same university, or at
no,
the?
no, no. He may be busy. He may be busy. He's in clinic.
Oh, okay.
He's
Yeah.
yeah.
Yeah, my brother, he just became a full neurosurgeon, but he's in Germany. So
Oh, he's in Germany. Okay.
yeah,
So I'm glad you loaded the articles. I think that's what I'm going to talk today. So, uh,
yeah,
so at least people can follow that, paper.
yeah. What we like about the recordings here on Clubhouse is that it stays interactive in a way. So when people click on this, they will still have the links available and can follow along in the chat what people ask and what you're responding to. Um, I like that about the recordings here on Clubhouse. So
Sure, sure.
that makes it. The only thing they cannot do is ask you a question personally. But other than that,
Yeah.
because we really have people around the world that are members,
Yeah.
so there's never a way to find a time that works for everyone. So yeah, the recordings are really helpful for people. So okay, we'll start in around 4 minutes. Thank you everyone for your patience, and yeah, it will be a really interesting talk of a very important subject, I think, around the world. So yeah, I'm really looking forward to this talk. Thank you so much for coming.
So I can start now.
It
There is 4 minutes still.
yeah, a few minutes are left, and then we'll start. Thank you.
Sure, sure.
Hi, Cami. Thank you for coming. How are you today?
We love you,
Thank you so much for
Cami.
inviting me. What an interesting topic.
Yes, meet Professor Pandey. Professor Pandey meets Kami. She's an amazing science journalist. She writes many amazing articles in major scientific journals. So, yeah, it's a pleasure. Thank you. Okay. Hi, Dennis. How are you? Thanks for coming. I think we can slowly start by introducing the audience, Professor Pandey. Welcome, everyone, to the Science Society, and of course, a special welcome to our guest speaker, Professor Dr. Pandey. And let me give you a little bit of information about him. So, he's Joseph Flathery, MD, endowed professor of psychiatry, the director of the Center for Alcohol Research and Epigenetics, professor in biochemistry and psychiatry and cell biology. He's the director of neuroscience alcoholism research, a senior VA career research scientist, and all at the University of Illinois at Chicago. And he's a nationally and internationally very well-known neuroscientist in alcohol addiction field, and he has contributed um, significantly towards a better understanding of the neurobiology of alcoholism. And he he won many awards. um, and his interest is to identify the molecular and epigenetic mechanisms of adolescents' alcohol exposure induced psychopathology in adulthood. And NIH has featured a lot of his work. Welcomes. So, we welcome you and we appreciate you being here. And before we start, we usually ask a couple of interview questions, if that's okay with you.
That's fine. That's fine. That's fine. Yeah.
Oh, perfect. So, what we usually ask is what, how come you ended up in the field of science or become a researcher? Was that always an interest of yours? Was there maybe a teacher or something specific that happened so you made your life in science? Thank you.
First, let me thank you so much for inviting me to really present some of our current work. And really, I am very pleased to be here. And thank you so much. Uh, my interest in the science started because I wanted to learn if there is disease, what is the mechanism and how the disease progresses. And particular area of the brain disorder, it just happened to that I started working in the brain when I joined my PhD, and then extended that to really better understand the psychiatric disorder, and in this case the addiction. uh, so this was my interest to begin with. When I was doing my master in biochemistry, I wanted to know more about the neurobiology of the brain disease. Thank you.
Oh, thank you so much. You did, because you made so many major contributions that will benefit so many people around the world. So um, thank you so much. And if you would like to give an introduction now about your recent or also about your work in general, feel free. And the stage is yours. Thank you.
Thanks again. Yeah, so my research interest is that uh, to understand the neurobiology of alcohol use disorder. Uh, and I think last 10 year I got interested because the very high prevalence of the adolescence binge drinking. Uh, and we know that during adolescence the brain is developing, uh, and then the genetic and epigenetic, genome and epigenome, they are very important. They shape the function of our body and the brain. So I thought that maybe if we can have a model where we can expose the developing brain to the alcohol and then see what happened to genetic and epigenetic changes, and if that has any impact on the adult psychopathology, such as depression, anxiety, or predisposition to the substance use and alcohol use disorder. And that's the reason I started that this problem of adolescents binge drinking is really huge, and it has a big impact on the individual life later on, and you will see that it has also a long lasting impact on the brain structural changes, also long lasting impact on the genetic and epigenetic changes, uh, and some of the behavior which we see developed during adolescence, they persist till adulthood. uh, so we felt that this is a very, very important area to investigate. Thank you. So can I start my talk?
Yes. Yes.
Continue.
Thank you so
Okay.
much. Yep.
So what I'm going to cover today, I think Katrina, she already has provided the link of this one recent publication from the lab, is targeted epigenomic editing ameliorate the adult anxiety and excessive drinking after adolescence alcohol exposure. Uh, so let me tell a little bit about uh, why adolescents binge drinking and the brain and epigenetics. Uh, I think I hope that you all know what the epigenome and epigenetics is, but just to give a brief idea that uh, the epigenetics in late term is the things above the genome. And so we know we have DNA sequence, and then DNA wrap around to important proteins, and those proteins are known as histone. And these histone, they get chemically modified. Uh, Even DNA also chemically get modified. So if you see the change in the DNA function or the gene function without changing the genetic code, uh, and because of the chemical modification of either itself DNA or the histone, uh, and that is the area the study is known as epigenetics mechanism. Uh, so we thought the epigenetics because it's very important developmentally. It really shaped the function of the developing cells, not only in the brain but other organs too. Uh, and we know that adolescence is very critical phase of the brain development. Uh, this is the time when brain goes neuromaturation, uh, changes in the neurotransmission, gene expression, uh, synaptic remodeling, where the neurons, they communicate each other, they make the connections, and this is the time when this process is really get mature. Uh, so and it's happened in several brain region. The brain is a very complex structure. It has nature has given several brain regions, and each brain regions carry the different function. uh, so this process of the neuromaturation occur in all brain regions. Some brain regions are very susceptible to the insult, uh, any kind of the trauma, in such case like alcohol exposure, and some brain regions are not. Uh, why adolescence? Because we know clinically, if someone is exposed to the alcohol at the age of 14 uh, or earlier, younger age, they are more than, I think, 5 to 7 times. They are more vulnerable to diagnose with the substance abuse disorder, psychiatric disorder later in life, compared to those individuals who start drinking at the age of 21. So this period of adolescence age is a very critical period for the brain maturation. And we thought that maybe the adolescents been drinking through the epigenetic changes, uh, causes the changes in the neurobiology, the neurochemistry of the brain, and thereby it changes the structure of the brain, synaptic connections, and how the neurons and various cells they communicate. And these changes may be in some brain region is permanent, and therefore those individuals they are more prone, are susceptible to develop the psychiatric disorders such as anxiety, or they develop the alcohol use disorder later in the life. And that is the reason we started. Now the questions against before us was that how we model the human adolescents binge drinking. And so there is a lot of preclinical study going on in the field, and so many contribution of the various researcher in the field. And it turned out to be that postnatal day in the animal model, in this case rat model, uh, from the 28 to 41 is very critical window, very much similar to somewhere junior high school to the early college, and this is the time the brain is really developing and a neuromaturation process going on. So what we thought that maybe we can give the binge-like exposure during this postnatal day 28 to 41, and then see what it has impact on the uh, epigenetic changes later in the life in adulthood, and also whether these in these animal model we see any behavioral phenotype of such as anxiety and alcohol drinking behavior. We focus on alcohol drinking behavior and anxiety because we know that alcohol is anti-anxiety compound. People they drink alcohol to self-medicate. Uh, when they drink, they feel relaxed, and because of the anti-anxiety property of the alcohol. So there is a very strong correlation between anxiety disorder and alcohol use disorder, and that is the reason we focus on these two behavioral phenotypes. So we use the animal model and we expose them with the binge-like drinking during critical phase of the adolescence, and then all behavioral phenotype and epigenetic changes were the measured at adulthood. So as I said that the normal epigenetic programming is required uh, from fetus to adolescence to adult for the proper brain development and proper functioning of the brain. Uh, but if something happen during adolescence or prenatal period, such as prenatal alcohol exposure or adolescence binge drinking or adolescence alcohol exposure, it changes the chromatin architecture of uh, uh, the epigenome. And when I say chromatin means that genetic material, gene, DNA, surrounded by the important protein and important regulatory protein, such as the gene transcription factors, the protein involved in regulating the gene expression. So this chromatin get condensed. When condensed means that the active portion of the DNA is not available uh, to for the transcription. So the gene expression or gene transcription is kind of arrested when the chromatin get condensed because the active portion of the DNA is not available. When chromatin is relaxed, open, that means that uh, the the important regulatory portion of the DNA of the gene is available for other protein to act and cause the transcription of the gene. Uh, so the gene is expressed more. So these chemical modifications they lead to the either condensed chromatin means they are decreasing the gene expression, or the open or relaxed chromatin that means they are uh, allowing there is a more gene expression. And so when there is a more histone acetylation, uh, then chromatin is open. When there is a less histone acetylation, then chromatin is condensed. Similarly, the repressive mark of the epigenome, depending upon what are the residue on the histone is getting methylated. Some of the methylation, in this case, which we have discussed in the paper, is STK27 methylation. If there is more, it also suppresses the condensed chromatin and the gene expression goes down. So we found that series of the work that adolescents binge drinking in the amygdala brain region, which is a center for anxiety, center for the motivational aspect of the drinking. It is also center of the fear. In this brain region, we found that chromatin goes to condensed chromatin architecture because of the decreased histone acetylation, because of the increased histone methylation, and also because of the increased DNA methylation. And these changes, they persist. So this condensed chromatin architecture in amygdala brain region, which is induced by the adolescent alcohol exposure, it persists till adulthood. And then we see the behavioral phenotype of anxiety and alcohol drinking behavior. These animals, they drink heavily at adulthood, and also they show the high level of anxiety-like behavior. Now, we also observed that the adolescent alcohol exposure in rat model, in animal model, causes a lot of the synaptic changes in the amygdala brain region. And these synaptic changes are, on the neurons, there is the spines, and these spines, they communicate each other through the synapse formation. And we found that adolescents binge drinking decreases their spines and also decreases the number of synapses in the amygdala brain region in adulthood. We also measure the several synaptic genes, and one of the synaptic genes is the activity regulated cytoskeleton associated protein known as ARC. And this synaptic gene ARC is very important. More ARC is good for the memory, and particularly when there is a memory formation is occurring in hippocampus, the ARC gene expression is more, and ARC play a very important role in the regulation of the memory development, memory formation, and also storage. Similarly, when ARC is less, the spines, the number is less, synapse is less, and we see that there is high anxiety like behavior. And also we have shown in different model that the animal, they consume a lot of alcohol, and because alcohol, when they drink, the ARC expression normalizes, it goes to the normal. So synaptic genes, several synaptic genes, some example is the ARC, which I talked, brain-derived neurotrophic factors, HOMER1, Neuro D1, D2, and also the synaptophysin, synaptotegmin. These are the genes I am taking a name. They are the very important synaptic genes, and it appears that all these genes, they get downregulated in adulthood after the adolescent alcohol exposure. But when we wanted to see that out of so many genes, synaptic genes which are altered in the amygdala, what are the important genes which may be where we can do the more detailed study? And it turned out to be that, and this is we call the gene network pathway. So we developed the synaptic gene network pathway in the amygdala brain circuitry. And what we observed that the ARC gene, ARC, is one of the hub genes. This gene is really interacting with several other synaptic genes like BDNF, like HOMER1, Neuro D1 and D2. A lot of these genes, they are being, the ARC gene is interacting with them. So we thought that maybe the ARC is a very important gene because it turned out to be a hub gene. And then maybe we can do a detailed study that how this gene is being regulated epigenetically. But before, so we investigated that, and we found that as we know that any gene has promoter, transcription start site, and then upstream to the promoter, we call the enhancer region. These enhancer regions are very important. They're upstream to the promoter. They interact with the promoter, and they regulate the gene expression. So we found that the chromatin architecture is condensed at the promoter of the ERK gene, as well as the enhancer region, which is 7 kb above the promoter. And there is also condensed chromatin, and that is due to the decreased histone acetylation, which is S3K27 acetylation, and increased S3K27 trimethylation. So these are the epigenetic marks suggesting that the chromatins are condensed at the enhancer region of the gene as well as at the promoter region of the gene. And there is a very good interaction, epigenetic interaction between the enhancer region and the promoter region, and the condensed chromatin leads to the decrease enhancer RNA. These enhancer RNA are very they are the small nucleotide, the 24 to 25 nucleotide. They are non-coding RNAs. They never translate into the protein, but they carry very important role. They interact with the transcription machinery at the promoter. So this ERK enhancer RNA, which is originating from the enhancer region, goes and bind to the promoter, and then there is a protein called negative elongation factor, NELF protein. If this protein NELF bind to the promoter, it suppresses the gene expression. But if you remove this NELF protein, negative elongation factor protein, from the promoter, then there is a gene expression, there is a more gene expression. So this enhancer RNA, which is coming out from the enhancer region of the ERK gene, it come and interact with the promoter and remove the NELF, and therefore the gene expression occur. But during adolescent alcohol exposure, this enhancer RNA expression gets suppressed because of the condensed chromatin at the enhancer region. And since enhancer region is suppressed, the promoter NELF is not being removed, and it leads to the decreased ERK expression. And this situation, and this is happening with the adolescent alcohol exposure in adulthood, and this condition we say high anxiety and also high alcohol drinking behavior in adulthood. So before we start a mechanistic or causal experiment, which the data is presented in this paper, we thought that maybe we can translate this in the human. So we had a cohort of the human postmortem amygdala, and this we obtained from University of Sydney, Australia. And these are the very well characterized brain bank, and we had a cohort where we have individual, they had the age of onset for alcohol use disorder early, before age of 21. And then we have a few cases where we have the age of onset after 21 year of the age. And what we observed that the people who started the drinking early, before age of 21, they have the suppressed ERK expression due to the condensed chromatin in their enhancer region. And also what we observed that the enhancer region, which I talk about the ERK gene, which is the above the promoter region, it's conserved along the species. So same enhancer region is also found in the human. And so we thought it's very good model where we can very nicely translate the animal finding to the human postmortem brain. And we found exactly the same finding, that early adolescence alcohol exposure in human amygdala also leads to the condensed chromatin due to the decreased histone acetylation and increased histone methylation at the enhancer region of the ERK gene, and therefore there is decreased ERK expression. And the people who started drinking after the age of 21, we do not see that such changes. That suggesting that maybe uh, the adolescential coil exposure leads to a permanent epigenetic reprogramming on certain gene, and that leads to their expression changes in adulthood and maybe relating to the phenotype of anxiety and drinking. So when we translated this into human postmortem brain, and then we thought that maybe we can go back to the animal model and try to correct the locus in the gene where they have condensed chromatin architecture, and that condensed chromatin architecture is at the enhancer region of the arc gene. So we identify a genomic locus of the abnormal epigenetic reprogramming. So we come up with this the tool and which is described in the paper is the CRISPR. And so the beauty of the CRISPR is that we can fuse the exogenous the protein, and this DCAS9 is really the mutated one. So so what it does that it does not have the catalytic activity, but the functional protein which get in the fuse with the DCAS9 in this case is the pre300, and that has the catalytic activity, and that can increase the histone acetylation where we can identify the genomic locus in the genome. And in this case, that what we did that we first developed the use the CRISPR Cas9 P300. P300 is an enzyme which really increases the histone acetylation. And then we developed the guided RNA and to guide the which locus of the gene which we would like to correct it with the CRISPR. And then for that purpose, we use the lentivirus, and these DCAS9 P300 and the guided RNA, they are packed in the lentivirus, and we first tested in the culture cells in PC12 cells, and we found that that DCAS9 P300 increases the histone acetylation in the culture cells, also increases the ERK expression, and we also verify that this is a very specific targeted effect because we chosen a lot of non-specific off-target gene predicted off-target, and we found they are not changed by the DCAS9 P300. So when we confirmed in PC12 cells, then we went back and we infused the lentivirus directly into the central nucleus of amygdala. This is the very specific region within the amygdala, which is very important, serve as a output for the amygdala brain region, and is also regulate the anxiety and drinking behavior. So we infuse the lentivirus of the DCAS9 containing DCAS9 P300 and the guided RNA into the central nucleus of amygdala. And after 2 weeks, we tested these animals for their anxiety and alcohol drinking behavior. And we got very excited when we saw the result that the infusion of the DCAS9 P300, which is the activatory CRISPR, which activates the histone acetylation, was able to attenuate the adolescence alcohol exposure, induce anxiety-like behavior, and also suppresses the alcohol drinking behavior. But it does not have effect on the sucrose drinking behavior. We wanted to have a control for the alcohol drinking behavior to also give these animals preference to drink the sucrose. And what we observed that they have decreased alcohol drinking behavior after infusion of the DCAS9 P300 with guided RNA for ERK enhancer region. Enhancer region we call here the synaptic activity response element because this is a very sensitive to the synaptic activity, and in acronym for this is the SARE, S-A-R-E. So the guided RNA was designed to this enhancer region known as stay aside. And by increasing the histone acetylation, and then we measured the histone acetylation in this region, and we found that the DCAS9 P300 also corrected the deficit in histone acetylation at the enhancer region of ERK gene, and also the ERK expression also get normalized. And it does not have any effect on the off target, the off gene target for the ERK guided RNA. So this experiment suggested that causally that the epigenetic abnormality at the enhancer region of the ERK really important in regulating the epigenetic changes as well as gene expression changes, ERK expression changes, as well as the behavioral phenotype of anxiety and drinking in adulthood. Now we went further, and what we wanted to see that how the epigenetic reprogramming at the enhancer region is regulating the function of the promoter of the gene, and that's the data all presented in the figure 2 of that paper. And so I was telling you all about the enhancer RNA, which is a non-coding RNA. And this enhancer RNA, when there is a more enhancer RNA, both plus minus strand, that means that enhancer region is very active. The adolescent alcohol exposure suppresses the enhancer RNA. But the enhancer RNA level get normalized by the DCAS9P300. So when we open the chromatin at the enhancer region due to the DCAS9P300, then these enhancer RNA, which were suppressed by the adolescence binge drinking in adulthood, they get normalized. And also I was telling you that there is a one protein NELF, which is negative elongation factor, that bind to the promoter. And when it bind to the promoter, it suppresses the gene expression. So after the adolescence alcohol exposure in adulthood, the NELF binding to the promoter is very high. But when we infuse the DCAS9P300, what we observe that enhancer region at the enhancer RNA goes up and it binds to the NELF and it removes the NELF from the promoter, and thereby the gene expression get normalized. Another observation we observed, we found in this experiment that remember we doing this targeted epigenomic editing at the enhancer region, which is the 7 kb above the promoter. We are not doing anything to the promoter of the gene. But when we measure the histone acetylation at the promoter region after the DCAS9 P300 infusion into the central amygdala, what we observe that histone acetylation is also going up at the promoter region. So we ask question why and how this is happening. And so we use a technique called this is what the people use in the field. They call the 3C assay, which is the chromatin capture assay. And in this assay, what you want to know is investigate that is the enhancer region is interacting with the promoter of the gene through the chromatin looping. So this chromatin, they turn around and this enhancer region interact with the promoter and thereby regulate the gene expression. And that's what we observed, that this interaction of the enhancer region with the promoter is really suppressed by the adolescent alcohol exposure. But when we did the editing at the enhancer region by DCAS9P300, we promote the interaction of the enhancer with the promoter, and thereby we remove the NELF from the promoter and we increase the gene expression. We suppress the anxiety and alcohol drinking behavior. So it was a very, very exciting and very, very fascinating to see that how a region of the gene, which is the far away from the promoter, through the epigenetic remodeling, interact with the promoter we call the chromatin looping, and thereby regulate the gene expression. So next question we ask in this, that if this is the case, then if we take a normal animal not exposed to the alcohol at all, and if we do a editing, and rather than activating the histone acetylation, if we can suppress the histone acetylation in normal animal at that locus of the gene, can we provoke the anxiety? Can we promote the alcohol drinking behavior? And that's what we observed. We took the negative inhibitory CRISPR, which is the DCAS9 crab, and what it does that it increases the ESTK27 trimethylation, which is a repressive mark of the epigenetic. So what we did that, same way we first construct this the virus and DCAS9 crab, and then guided RNA for the enhancer region of the arc. We packed them together in lentivirus and first tested in the PC12 cells, and we found that in PC12 cells this DCAS9 crab suppresses the arc expression because of the increased ESTK27 trimethylation. It does not have any effect on the off target, the gene target, and therefore then we took that construct of the lentivirus and we infused in the central nucleus of amygdala of the control animal, alcohol naive animal. They are not exposed to the alcohol, and these are the adult rats. And what we observed that after 2 weeks, these rats, they show high anxiety like behavior, very much similar to the adolescence alcohol exposed animal. And also these animals, they drink heavily. They drink a lot more compared to the control animal. And their drinking is like very much similar to like adolescence alcohol exposure, the induced drinking behavior. So we mimic the phenotype of adolescence alcohol exposure in normal animal after depositing more repressive mark of the epigenetic, which is the ESTK27 trimethylation, at the enhancer region of the ARC gene, and suppresses the ARC expression. We do not see any effect on the sucrose drinking behavior, which was used as a control. And again, we wanted to know whether the condensed chromatin architecture at the enhancer region due to the increased histone acetylation, ESTK27 trimethylation, there is a decrease in enhancer RNA. And so we observed that when we have the condensed chromatin at the enhancer region share side, we have decreased enhancer RNA. And when we have decreased enhancer RNA, then we have more NELF binding. The protein I was telling you all that, the negative elongation factor, which bind to the promoter of the gene. So what we observed that the NELF binding at the promoter is more, and therefore the ARC expression is shut down. Because of the decreased histone the acetylation and increased histone methylation, ESTK27, at the enhancer region of the ARC gene, there is no chromatin looping is occurring. We do not observe any interaction of the enhancer with the promoter, and one of the reason is that because enhancer RNA is suppressed, level is suppressed, and looks like also there is no chromatin looping is occurring. So to sum up this whole study, which came in the Science Advances in the month of May, that adolescence alcohol exposure causes the epigenetic reprogramming, and it in such a way that chromatin get in condensed chromatin architecture due to the increased histone acetylation, and due to the decreased histone acetylation and increased histone ESTK27 trimethylation, which is the repressive mark. And as a result, there are the suppression of the enhancer RNA, and enhancer and promoter, they may not interacting, and the NELF is bind more to the promoter and decreases the ARC expression and provoke the anxiety and alcohol drinking behavior. if we use the DCAS9 P300 and functionally increase the histone acetylation at the enhancer region of the gene, we see there is a chromatin the chromatin looping is occurring. Enhancer region interact with the promoter, and also there is a more enhancer RNA, and it remove the null from the promoter, and there is a more ERK expression. ERK expression is get normalized, which were suppressed by the adolescent alcohol exposure, and therefore the phenotype of anxiety and alcohol drinking behavior is attenuated. We can really mimic these responses by using the inhibitory CRISPR. That means that we increases the repressive mark STK37 trimethylation at the enhancer region of the gene, and therefore we shut down the gene expression, and then animal they become anxious and they consume more alcohol. So that was the overall discovery for this paper, and this is for the first time demonstrated that a targeted epigenomic editing, which is a functional epigenetic editing, that really the normalizes the suppression of the gene expression, and also attenuated the anxiety and alcohol drinking behavior. So a lot of people really contributed to this work. It's a teamwork and various expertise, and creating the CRISPR tool and the animal model, and then conducting the research in the human postmortem brain, and also the fundings from the NIH and from the other funding agency is really help us to carry out this important study for the field. And with that, I will stop, and thank you all very much. And I'll take questions, and maybe we can start a discussion on this work. Thank you all so much.
Oh, thank you, Professor. This was an amazing talk, and congratulations for this really thorough work that helps us understand so much about these mechanisms of alcohol use disorders. So, yeah, we really appreciate it. And I wanted to ask, maybe in the future, are you maybe planning to check if with the CRISPR treatment, basically, or do you see a transgenerational effect of alcohol use disorder, like exposure, that basically the next generation are have a higher vulnerability, and maybe also changes in ARC, and with the treatment in the parents that were kind of exposed to alcohol during development, would they also maybe be protected? Like, would this treatment maybe protect future generations also?
Very, very nice question, and I think that's where the future is. First thing is that how the epigenomic editing can bring to the human population. I think a lot of work need to be done and need to be evaluated about the how these viruses, and whether the lentivirus is stable, or maybe we have to focus on the adenoviruses. So I think the field is progressing very well, and for some other disease. So there is a hope that maybe one day, if this comes into the human, and maybe we can able to correct the deficit induced by the adolescent drinking. Your first question was that whether there's a transgenerational effect of this. Yes, there are a lot of study going on in the field where they have done with the prenatal alcohol exposure, which has impact on the babies, and particularly on the DNA methylation and of certain genes, and those genes are the hypermethylated state of the gene is going from generation 1 to generation 2 to generation 3. So I think these epigenetic modifications are some of the modifications are going from one generation to other generation, and it appears that they play a very important role in regulating the behavioral phenotype. So I think this area is really of medicine is very, very fascinating, and a lot more work need to be done. But at least we now understanding that, 10 year back, 12 year back, this epigenome was a very hot area in the cancer field. But now in the neuroscience, and particularly understanding the brain disease, now it's developing very nicely. And we know now that in this case, like alcohol, drinking really causes some long-lasting impact on the epigenome. And also, not our work, but other scientists in the field very nicely demonstrated that some of the epigenetic changes which occur due to the prenatal exposure, particularly in the fetal alcohol spectrum disorder (FSD) area, there is a transgenerational impact, and some of the modifications are going from next generation. Thank you.
Yeah, thank you so much. Joyce, please go ahead.
Okay. I was wondering, as I understand it, alcohol consumption causes increased intestinal permeability, which then causes the ability for more microbes to cross and would change the gut flora and so on. Do you think that this might have something to do with the epigenetic effects and even the inheritable? I mean, if the microbiome is changed, the child grows up in an environment with a different microbiome in the house, which they acquire, or they acquire through their, close contact with the parent, or even when they're going through the vaginal canal in being born. Thanks, I'm done.
Yeah, I think the very, very nice question, and I think this microbiome area is really very fascinating area, and we know that the alcohol drinking caused the gut to leak, so definitely I think the microbiome do get impacted by the alcohol drinking, and 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 of course that may lead to the epigenetic changes. The question is that how much is microbiome mediated changes are occurring directly into the brain. But one of the system, neuroimmune system, which really impacted both in periphery as well as in the brain, uh, it appears to be related to microbiome as well as the epigenetic changes. So, it's very hard to tell that what really causing or mediating, but if we know at least some abnormality on some genomic locus, and if you try to correct that and then see phenotype is get attenuated, I think that's what the target is right now. There may be multiple factors which may be induced by the alcohol drinking, and they may lead to the biological changes. But of course, the microbiome is one of the very emerging field, and I think we will know a lot more in the future. And currently, a lot of investigators they are working in that area.
Thank you very much.
Thank you.
Ashkash, did you have a question? I hope I'm saying your name right.
Yeah, thank you. I don't have much actually. Thank you, Dr. Pandey, for the nice talk. Thank you very much.
Thanks, Akash.
Thank you. Fatih, I hope I'm saying your name right. Do you have a question? Maybe he's away from the phone right now. Suraf and Dr. Olu, please go ahead if you have a question.
Oh no,
Dr.
I'm actually
Pandey.
here. Thank you. I was
All
just
right,
trying to
please
figure
go
out
ahead.
them. Oh no,
Go
no,
ahead.
Saurav, if you want to go ahead,
Fatih,
please.
go ahead.
That's fine.
No. You go ahead. I'll take the next.
I know
Next.
it was a
Please.
very informative talk, and discussion. I mean, there were some technicality that I couldn't understand, but absolutely it was a very informative topic, Dr. Pandey, and I actually I'm curious if your studies have anything to do with paranoid schizophrenia psychologically, and if there was any work done to it. But yeah, other than that, I mean, it was really good. Thank you. I really appreciate it.
No, thanks, Saurabh. I think, yeah. I think my group here at Department of Psychiatry in the neuroscience group, uh, we have done a lot of work in the schizophrenia also, and what we observed that the same kind of epigenetic modifications do occur also in schizophrenia too. So there are studies being done in the depression field, in schizophrenia, in bipolar, substance use, and alcohol use disorder. Uh, the basic fundamental question is that these brain disease, they have abnormal epigenetic reprogramming. The question is that what kind of gene network pathway they get affected. Is there is a common pathway getting affected in all these brain diseases? Are certain network pathways very specific to the certain brain disorder? But fundamentally, what we are seeing that this DNA methylation, histone modifications, uh, they are occurring across the various brain diseases, whether it is schizophrenia, depression, bipolar, or addictive behavior.
Great. Thank you, Dr. Pandey.
Thanks.
Dr. Pandey, this is Saurav. First of all, thank you. It is hugely, for people who may not be directly into the research, for them it's going to be a lot to grasp, but it's brilliant piece of, enlightenment that you took us through, and thank you for that. I'm sure there'll be a lot of work happening in the future too, and we'll hear more about it, and we'll be reaching towards better and faster cure to these ailments. Saying that, I just wanted to kind of just curious about to figuring out whether anything to do with ethnicity has any impact to this, because there are some studies which says alcoholic ability to can adhere or manage alcohol. There are some races or some ethnicity has a better one than the others. So I mean, is there any link to this? It's completely an outside question, but I thought I'll just park it here.
No, I think it's a very nice question, and I think uh, one thing is very important when we talk the gene and environmental environment. I think that's where the epigenome comes and play a very important role. Uh, and simple example I can give to you that suppose if some gene has uh, the gene polymorphism on certain gene, and we know that that gene is really important in predisposing or developing a disease, uh, say for example the alcohol use disorder. And there are studies indicating that that gene polymorphism in some country is really causing the disease like alcohol use disorder, but in other country that polymorphism is protecting to develop the alcohol use disorder. So when that question, when that things eh, comes in our mind, it definitely come that maybe the epigenetic modifications are important here, because of the different environment in different countries, the weather and temperature and food and so many things together, maybe shaping the epigenome in different way, such a way that some genetic polymorphism is not predisposing that disease. Whereas in some country or some ethnicity group that the environment is such a way that it's changing the epigenome such a way that polymorphism get active and then lead to the predisposition of the disease. So it is important, and I think it's all depends upon our surrounding, uh, and that may have impact on shaping the our epigenome and thereby genome and then function of the various organ of our body. So that's what I can say.
Thank you, Dr. Pandey. I'm done.
Dr. Olu, would you like to ask your question?
I just actually want to say hi. If I I could actually go
Hi, Olu, how are
downstairs.
you?
Good. I could go downstairs if I had specific questions, but
Thanks for encouraging me to join this group.
yeah,
Thank you.
no, it's a great group, a great. They have wonderful science talks, so
I think
I'm
so.
glad you're able to do it.
Thank
I do have sort of a general question, thinking about your very cool use of CRISPR, and then thinking about possible, therapeutic developments that can come from your work. Have you thought about? I mean, I think it's probably not feasible or realistic to think about CRISPR as a treatment, but maybe have you thought about other types of treatments that might target the same genes that were edited?
Yeah, I think that's a very good question, Olu. So, uh, as you know that we are focusing, and also we publish that. Uh, so there is a way some of the drug being used in the cancer field. Uh, so one drug is known as SAHA. This is a FDA approved drug for the treatment of cutaneous lymphoma. This is the histone deacetylase inhibitors, and what it does that it inhibits the histone deacetylase. Therefore, the acyl group they're not removed from the histone, and you increase the histone acetylation, and therefore you open the chromatin. Uh, so I think we tested that compound in our model, and we found that the SDEC inhibitors is really able to attenuate alcohol drinking behavior and also attenuate the anxiety behavior. Uh, and since it is FDA approved drug, I think it has high potential to bring that into clinical area and do some small scale clinical trials on that. And the drug is the DNA methyltransferase inhibitors, and that drug also has ability to open the chromatin due to the inhibition of DNA methylation. And we tested that also in our animal model, and we found that DNA methylation inhibitor is also able to attenuate anxiety and alcohol drinking behavior. So pharmacologically, I think there are the drug which really has ability to manipulate the epigenome and thereby the behavioral phenotype. Uh, but they need to bring to the clinical population in terms of maybe small scale of the clinical trial and see what dose of the drug is effective, because these are the cancer drug, and basically they're being used in the cancer terminally ill patient, and they use very high dose of the drug and there is some side effect. uh, so there's a lot of effort going on in the field that maybe manipulating those small molecule such a way that they may have a specific impact on the targeted the enzyme, and thereby we can reduce their side effect. Uh, so I think a lot of effort is going towards developing the small molecules, and we are also in going in that direction.
Very cool. Thanks, Subhash.
Thank you, Olu.
Yeah, and thank you, Dr. Olu, for always giving so nice recommendations. We really appreciate your support. And yeah, that's really interesting to hear that methylation inhibitors can also, because there are a few interesting ones that are small compounds that maybe could pass the blood-brain area and are less toxic than
Yeah.
others from cancer research. So that's really exciting. So
Also,
yeah.
there is also the histone acyl transferase activators. So they are directly able to increases the histone acetylation. That the drug compound has been developed, being tested in the animal model for the learning and memory, and in neurodegenerative disease model, that compound activator of the HAT, we call HAT histone acyl transferase activator, has ability to correct the deficit in learning and memory. So that compound also has high potential to bring the cases where we using the DCAS9 pre-300 to see that whether the HAT activator is also effective in similar way as the CRISPR approach for the gene. Only the thing is that this compound, they may have very wide effect and not the targeted effect. So that's where the effort has to be done, that if we identify a certain locus which are abnormal, and then see whether that locus also get normalized by the use of the pharmacological agent such as HIT inhibitors, uh, the SDAC inhibitors, DNMT inhibitors, or HIT activators. Thank you.
Yes, thank you. And Victoria had the question in the chat. If you saw also in adult differences in impulsive behavior in the rats.
So in adult animal, you mean?
Yes.
What the question?
Yes.
Okay.
To tell.
So we do also see the same genes also get impacted by the adult alcohol exposure, uh, and also we found that in adult also these genes regulate the anxiety and alcohol drinking behavior. Uh, Only difference is that if the exposure is done during adolescence phase, when the brain is undergoing neuromaturation, these effect is long lasting. Whereas in adult, uh, these effect may be easily reversible and maybe not that long lasting as in the adolescence. So there is a lot of study going on in the field. Uh, we have a consortium known as the Neurobiology of Adolescence Drinking and Adulthood, NADIA. And so that's where we're doing a lot of study where we try to see that the changes induced by the adolescent alcohol exposure, uh, it appears that they are long lasting. But to answer the question, yes, similar change do occur with adult alcohol exposure, and it appears that both the ARC as well as other gene, which is the brain-derived neurotrophic factors, uh, they are decreased by the adult alcohol exposure during withdrawal, and we also have shown that they play important role in regulating the development of anxiety-like withdrawal symptoms during early alcohol exposure, early withdrawal after chronic alcohol exposure. Uh, so it changes the how long those changes persist. That's where that it varies. That so when brain is developing, the changes, there is already epigenetic reprogramming occur, and some of the reprogrammed epigenome they persist. Whereas in adult, maybe it's not that long lasting. Thank you.
Yes, thank you so much. And yeah, we are going for an hour, so I know you you have a very busy schedule. So I want to thank you so much for answering all of our questions and for giving this amazing talk. And thank you for doing the work you're doing, which will help so many people. And we really appreciate it. So thank you so much. And thank you, Gabi, for helping. We really appreciate it. Thank you.
Thank you. Thank you so much for this opportunity. And it was really my pleasure to give this talk. Thanks a lot.
Yeah, wonderful. Maybe one day like next year or so. That would be wonderful.
Yeah. There is a lot of development going on. I'm happy to share the new development in the field.
That would be wonderful. Okay.
Thank you.
Closing the room. Thank you everyone for coming and asking great questions, and um, we really appreciate it. And yeah, enjoy the rest of your day, and hear you all back soon.
Thank
Thanks.
you. Thanks so much. Bye.
Bye. Close the room in 3, 2, 1. Bye everyone. Thank you.